Question number 1. In eukaryotic cell, formylated tRNA is important for initiation of translation. Cyclohexamide block the elongation during translation. Cytosolic ribosomes are smaller than those found in prokaryotes. Erythromycin inhibit elongation during translation. Correct answer is option B. In eukaryotic cyclohexamide block elongation during translation. Question 2. The mushroom poison amanitin is the inhibitor of protein synthesis, mRNA synthesis, DNA synthesis, adenosine synthesis. Correct answer is option B, mRNA synthesis. Question 3. Tetracycline prevents synthesis of polypeptide by blocking mRNA formation from DNA, releasing peptides from mRNA and tRNA complex, competing with mRNA for ribosomal binding sites, preventing binding of amino acid tRNA. Correct answer is option D. Tetracycline prevents synthesis of polypeptide by preventing binding of amino acid tRNA. Question 4. In prokaryotes, chloramphenicol cause premature release of the polypeptide chain, cause misreading of the mRNA, depolymerize DNA, inhibit peptidal transferase activity. Correct answer is option A, cause premature release of polypeptide chain. Question 5. Streptomycin prevents synthesis of polypeptide by inhibiting initiation process, releasing premature polypeptide, inhibiting peptidal transferase activity, inhibiting translocation. Correct answer is option A. Streptomycin prevents synthesis of polypeptide by inhibiting initiation process. Question 6. Erythromycin acts on ribosome and inhibit formation of initiation complex, binding of amino acid tRNA, peptidal transferase activity, translocation. Correct answer is option D, translocation. Erythromycin act on ribosome and inhibit translocation. Question 7. The binding of prokaryotic DNA-dependent RNA polymerase to promoter site of the gene is inhibited by the antibiotic puromycin, rifamycin, terramycin, streptomycin. Correct answer is option C. Terramycin is the antibiotic. Question 8. The gene which is transcribed during repressor is structural, regulator, promoter, operator. Correct answer is option B, is the regulator gene. Question 9. The gene of lac operon which has constitutive expression is ICPZ. Correct answer is option A. Question number 10. The minimum effective size of an operator for lac repressor binding is 5 base pair, 10 base pair, 15 base pair, 17 base pair. Correct answer is option D, 17 base pair. 11. To common structural gene transcription, the region which should be free on lac operon is promoter site, operator locus, Y gene, A gene. Correct answer is option B, operator locus. Question number 12. In the lac operon concept, a protein molecule is operator, inducer, promoter, repressor. Correct answer is option D, is a repressor. Question number 30. The catabolite repression is mediated by a catabolite gene activator protein in conjugation with AMP, GMP, CMP, CGMP. Correct answer is option C, CAMP. Question 40. The enzyme DNA ligase introduce superhelical twists, connect the end of two DNA chains, unwind the double helix, synthesis RNA primer. Correct answer is option B, connects the end of two DNA chain. 50. Restriction endonuclease cut RNA chain at specific location. Excise intron from hnRNA. Remove Okazaki fragment. Act as a diffusive enzyme to protect the host bacterial DNA from DNA of foreign organism. Correct answer is option D. Act as a defensive enzyme to protect host bacterial DNA from DNA of foreign organism. 60. The most likely lethal mutation is substitution of adenine for cytosine, insertion of one nucleotide, deletion of three nucleotides, substitution of cytosine for guanine. Correct answer is option B. Insertion of one nucleotide and deletion of three nucleotides. 70. Restriction endonuclease recognize and cut a certain sequence of single-strand DNA, double-stranded DNA, RNA and protein. Correct answer is option B, double-stranded DNA. 80. Positive control of induction is the best described as a control system in which an operand functions unless it is switched off by a repressor protein. Only after a repressor protein is inactivated by an inducer. Only after an inducer protein which can be inactivated by co-repressor switch it on. Only after inducer protein which is activated by inducer switch it on. Correct answer is option D. Only after inducer protein which is activated by inducer switch it on. 90. Interferon is a virus, is a bacterial product, is a synthetic antiviral agent, require expression of cellular gene. Correct answer is option D. Require expression of cellular gene. Question number 20. Repressor bind to DNA sequence and regulate transcription. This sequence is called attenuated, termination, anti-termination and operator. Correct answer is option D. Operator. Okazaki fragment is related to DNA synthesis, protein synthesis, mRNA formation, tRNA formation. Correct answer is option A. Okazaki fragment is related to DNA synthesis. 22. The region of DNA known as Tata box is the site for binding of DNA polymerase, DNA topoisomerase, DNA dependent RNA polymerase, polynucleotide phosphorylase. Correct answer is option C. DNA dependent RNA polymerase. Question number 23. Reverse transcriptase is capable of synthesizing RNA to DNA, DNA to RNA, RNA to RNA, DNA to DNA. Correct answer is option B. Correct answer is option A. RNA to DNA. Or tetrovirus is poliovirus, HIV, herpes virus, tobacco mosaic virus. Correct answer is option B. Tetrovirus is HIV. 25. Peptidal transferase activity is located in elongation factor, a charged tRNA molecule, ribosomal protein, a soluble cytosolic protein. Correct answer is option C. Ribosomal protein. 26. Ultraviolet light can damage a DNA strand causing two adjacent purine residue to form covalently bounded dimer. Two adjacent pyrimidine residue to form covalently bounded dimer. Distribution of phosphodiester linkage. Distribution of non-covalent linkage. Correct answer is option B. Two adjacent pyrimidine dimer residue to form covalent bounded dimer. 27. Defective enzyme in Hurler syndrome is alpha L diuronidase iod. Correct answer is option A, alpha L diurodinase. 28. Presence of arginine can be detected by Sakaguchi reaction, Millin's reaction, Hopkins-Cole reaction, gas chromatography. Correct answer is option A, Sakaguchi reaction. 29. A nitrogenous base that does not occur in mRNA is cytosine, thymine, uracil, and all of these. Correct answer is option C. Cytosine is the nitrogen base that does not occur in mRNA. 30. In nucleotides, phosphate is attached to sugar by salt bond, hydrogen bond, ester or glycosidic bond. Correct answer is ester bond. So in nucleotides, phosphate is attached to sugar by ester bond.